All right, I'm gonna turn the recording back on. Unlike I did in class the other night. All right, do you have some specific questions to start with, or?
So there was a lot in, I don't remember any of the specific questions, but on chapter 4, during the homework assignments, I just didn't feel super knowledgeable on that chapter specifically.
Yeah. That's I think we didn't spend a lot of time on the details in class, if I remember correctly, because we kind of ran out of time with the other chapter. But let's see, let me just pull up that PowerPoint. Would be chapter 4. And I'll go ahead and share this just so that we kind of walk through. I think maybe
oh, go ahead.
I was going to say maybe a good way to approach it would be for me to pull up the study guide also.
Oh, yeah.
And then start with those specific things that are listed on the study guide, and then we can
From there.
cover other things that come up as we're going through it.
Okay. Let me see.
Come on, computer. I need to clean my computer again. I just cleaned it last. I guess I need to clean it every week. It's just so slow. It doesn't even want to open Word. That's absurd. That
Love what you did there. Yeah, I was just gonna say, I apologize in advance. I'm sure that the dog is going to bark, and he's so loud when he does.
Oh, no worries. Oh, we've got a little. My boyfriend's dachshund is old, and he's probably not going to make it through the day. So we're kind of.
Oh, I'm sorry to hear that.
He collapsed the morning of Keith, my boyfriend's surgery. I was like, God, please don't die today. We can't.
Right.
Enough going on.
Yikes. That does sound like a lot. Is your boyfriend doing well?
He is. He's doing well. Wait, unfortunately, his surgery ended up being so late that the doctor left and didn't tell us how it went. And he's got an appointment in 2 weeks. So we got to wait 2 weeks to find out.
Lovely.
I know, it's kind of like great. And the doctor told me he was going to call me when he finished his procedure, but he didn't call me. And I was like, great, thanks.
I'd be calling him. I'd be like, listen.
So, but the nurses told him it went well. So hopefully,
oh, that's good.
It was a well, it didn't matter. We don't need to share the details. He always tells me I go into share too much detail. I'm like,
share too much.
I forget.
I've been told that a few times too. I get it. It's okay.
All right. So I've got, let me see here. Do you have your study guide pulled up? Or
I have my notes pulled up. I wanted to pull up some notes, or I wanted to take some notes.
Okay, yeah.
From the session here.
So I'll share it for now, and then I'll probably just stick to the PowerPoint instead of going back and forth.
There we go.
My closed captioning every time I change a share. Okay, so the functions of the listed organelles, and then I have detailed on there the specific ones, which is pretty much all of them. The cell membrane, cytoplasm, DNA, ribosomes, RNA. So you do need to know the 3 types of RNA and where they function. I can't remember if in this class we have a different a separate chapter on protein synthesis. Usually we do, but I can't remember if it's separate in here. So that might be covered in more detail later. So I might need to go into a little more detail on that. And then lysosomes, mitochondria, Golgi, rough ER, smooth ER. And I put in there many pages and slides. You find them.
Mhm.
Basically is what I said there. ATP and where it's produced, which kind of overlaps into chapter 5. So that was also information that was on the other chapter 5 class activity that we did in the discussion board. And then chemical groups DNA is made of. Oh, that's interesting. Okay, so that is a bullet that I have on there. So we do cover it in a later chapter. So for some reason, there's a question on there about that, even though that detail is not in chapter 4 or any of the chapters that we're going to have covered before the quiz. So that's why I have that link to chapter 3. So we skipped chapters 2 and 3, but I guess they're putting questions on there from them, which is not my favorite thing that they do. Oh, and then there's also some additional different organelles and bacteria that you need to be familiar with. Some of them are the same, but there are some that are different. And then domains, groupings, binomial naming, which I think we kind of went over that a little bit in chapter 1 also. So let's start with the organelles and go in order of what it shows in the PowerPoint. All right, so there aren't any questions about the microscopes. We can skip all of that part. So the plasma membrane, this is also covered in chapter 5, where we went into more detail of the different types of transport across the phospholipid bilayer, the plasma membrane. Do you feel comfortable with that structure of that, or do you want me to go into more detail on?
If you could go over it briefly, that would be
Sure.
good.
So I'm going to just use the picture for this one because I think it kind of makes more sense when we talk about it. So the phospholipids, and this is another, the details of phospholipids were covered in one of the chemistry chapters, 2 or 3. So that's kind of might be why that's a little confusing. But phospholipids they're called amphipathic. Oh, goodness gracious. Now the annotation toolbar is different because it updated my great, everything's totally different. Perfect. And lovely. Phospholipids are called amphipathic. I'm just gonna write it up here. Because of the fact that it's kind of like if you think of ambidextrous, if somebody can use their right or left hand in order to do something, that's how the phospholipids are called. And that's because it has one end, the polar head that you see here on the top is, actually, I'm going to do this the other way here. Makes more sense to do it like this. So amphipathic. The polar head is hydrophilic, which means water liking or water loving. And then the non-polar tail down here, let's put it down here since there's another word right next to there.
Hydrophilic, which means water liking.
Exactly. And then the non-polar tail is hydrophobic, which means water fearing or does not like water, does not dissolve in water. Most lipids in general are hydrophobic, the whole entire thing. If you think about a vinaigrette salad dressing, the oil that's the lipid part separates out from the aqueous part that has the water and the vinegar in it. And, you know, when you put it on your salad, if you don't want to have just straight oil on your salad, you have to shake it up to get it to mix together, and then it'll separate again. So most lipids are hydrophobic. So the phospholipids are unique in that they are amphipathic, where they have one end that's hydrophobic and another one that is hydrophilic. Where's the box? Oh, here we go. Yeah, they totally changed the annotation. You know, I hate it when they change stuff. Leave it alone. So phospholipids have the polar head, which is the hydrophilic part that likes water. And that is a unique property of lipids. And then the fatty acid tails are what form the nonpolar part of it that is calling the nonpolar tail, are hydrophobic. And those are trying to get away from water. So this is the basis for how the phospholipid bilayer forms. And it's called a bilayer because there's two layers. So if you look at the one that I put a circle around here, there's one layer there on top, and then the other layer is on the bottom, and it's facing the opposite direction. So those non-polar tails are facing towards each other because they're trying to get away from water.
Okay.
There's mostly water outside of the cell where you have extracellular fluid, and there's mostly water inside of the cell where you have your cytoplasm. And so that is the basis for why the phospholipids orient the way that they do, and then also the fact that they are semi-permeable, meaning that only certain things can pass through them. So because of this charge difference, the non-polar tails, the hydrophobic part, they don't like anything that's charged. And so that's why nothing that's charged can pass through between the phospholipids. And that's why larger things need those protein channels to transport through. So non-polar, very small compounds can freely pass through the membrane. That's why we talked about the oxygen and carbon dioxide can freely diffuse across the phospholipid bilayer. And it is literally just going right between those little phospholipids for oxygen and carbon dioxide. They can just diffuse right through the field. There aren't any bonds that are holding these phospholipids to each other. They're just basically floating. So sometimes you'll see it referred to as a fluid mosaic model, because the only thing that's keeping them situated the way that they are is that those hydrophobic tails are trying to stay away from the water, and the hydrophilic heads are liking to be towards the water. So it's pretty crazy, I think, how it's set up, in that, or how it functions, and that there's not really any bonds holding everything together.
Yeah, that is really cool.
So we'll leave that at that for now, and then I'll see if you have any other questions that come up as we go through the, how do I get rid of, okay, clear, here we go. Everything is different now. Clear all my drawings. Maybe I'll save this one real quick. I don't know if you saw that I posted a lot of additional material in the Quiz 1 study guide announcement, and sent some of it in the Canvas inbox, but I posted a lot more in the announcement, so I might add this to there as well. And I'll probably add the recording of this video also. My computer's being stupid. All right, so let me do that before I clear it. All right, and so that's the different proteins that we talked about that are embedded within the membrane. They function differently to regulate what can pass across the membrane, and then also some of them as receptors that are kind of like the substrate here would be a key fitting into the lock to tell some cascade of events to happen inside of the cell. So that's the way that our cells communicate and know what they need to do to deal with the environment and what's going on outside of the cell. Some of them hold two cells together, so that would be different types of junctions between them. All right, and then I think there's anything on this. Let me pull the study guide up real quick. Okay, yeah, on this one, there's nothing specific about the cell theory. So I'm just going to kind of keep going on that one. It did say know the difference between the prokaryotes and eukaryotes. So the eukaryotes, in addition to that plasma membrane, there's a second phospholipid bilayer membrane that forms the nuclear membrane that separates the DNA from the cytoplasm, whereas prokaryotic cells don't have that. They just have their DNA is just kind of floating in the middle of the cell. So, and I forgot, it starts on prokaryotes here. So we'll go with this one, start with this one first. The biggest and the ones that we talk about the most as far as prokaryotic cells are going to be the bacteria. And that's because those are the ones that we have a lot of bacterial infections that can make us sick. And so we're more interested in the things that can make us sick. Archaea are also prokaryotes. They're considered extremophiles because they can live, a lot of them, in extreme environments like extreme temperature or extreme pH. There are no known diseases caused by archaea. And they're also considered to be archaic, if that helps you remember, the oldest organisms, living organisms. So structures in the bacterial cell would be that they still have that same plasma membrane. Plasma membrane is interchangeable with cell membrane. So if you see either of those terms, it means the same thing. Plasma membrane or cell membrane are both that phospholipid bilayer that we just looked at. So they still have that phospholipid bilayer, but then they also have an additional cell wall that is located outside of that cell membrane that provides them extra protection. So cell wall and cell membrane are two different things. Animal cells do not have a cell wall. And then, here it says plasma membrane is the same as eukaryotes. Some bacteria also have a capsule, which is an additional protective layer outside of the cell wall. So I don't think you need to know the details of the compounds that make up the cell wall versus the capsule, but those are basically two different levels of protection outside of the plasma membrane that bacteria have. Not all bacteria have a capsule, but most bacteria, in fact, not all bacteria have a cell wall, but most bacteria have a cell wall. They do have DNA. It is the genetic material for the bacteria. It's the same chemical structure as the DNA in our cells, in eukaryotic cells. But we have multiple linear chromosomes, which we'll learn about later. They only have a single circular chromosome. So they just have one, whereas we have 46 chromosomes. And they are, like I said, just kind of floating in the cytoplasm. They're not in an enclosed membrane. The region where the DNA is found in a bacterial cell or prokaryote is called the nucleoid region. They do also have ribosomes, which we're going to learn a lot more later about, but ribosomes are the organelles that carry out protein synthesis. So that's important. And then these other appendages, these are some of the ones that you definitely need to know for quiz purposes that are bacterial specific structures. So flagella are those tail-like structures for propulsion. Fimbriae are kind of finger-like projections that allow them to attach to surfaces. And then conjugation pili are a really important one because these are what allows different bacteria to transfer antibiotic resistance, and they don't even have to be the same species. So they can actually share genetic material with I like to say a non-related bacteria. So it's not their child, it's not their brother, sister, mother, like they don't have those, but it can just some random bacteria that can receive this attachment by the conjugation pili can share genetic material, and the biggest outcome of that is antibiotic resistance. So they can share their ability to exist in the presence of an antibiotic that would normally kill that bacteria. Why did it do that? Put it in the middle of the word. Share genetic. Oh, goodness gracious. And I can't remember whether it is in this class that it does it, or if it was in a different class that I had it in, but the conjugation pili are sometimes called the fertility pili, which I'm not really sure why they call it that, because it's not that they're replicating. It's not that they're having offspring. It's just that they're sharing part of the genetic information. So I don't know why they call it that, but sometimes you might also see that genetic material that's shared is, it can be chromosomal. I think it can be part of the chromosome, but usually it's going to be plasmid DNA, which is a smaller single separate circle of DNA. So the bacteria that can share via a conjugation pili have that F plasmid, and they can actually share that F plasmid with bacteria that don't have the capability to form the conjugation pili, so that they can then acquire the ability to share genetic material with adjacent bacteria. Sneaky little guys. But that's definitely one that I remember a lot of students missing the question that was related to that one session. So that's why I'm spending a little bit of time on that one.
I appreciate it.
Sometimes those little bullet points at the bottom of the slide don't make it into people's consciousness. And then you're like, ah, I didn't know you're going to get that detailed on the question. And there aren't any pictures. I wish they would have some, because I think it helps students remember what the different structures are. But if it helps you to look at the pictures, these are the different structures that we just talked about in the picture.
Okay.
Here. So if that helps you remember it. All right, so those are the main structures from the prokaryotic cells. And then the eukaryotic cells, there's 4 different kingdoms that have eukaryote, or that are in the domain eukarya, which means that they have eukaryotic cells. And these are the protists, fungi, plants, and animals. I think the only specific ones that you need to know are the bacteria and archaea. You do need to know that those kingdoms are included as eukaryotes, but as far as the organelles and structures, we're basically going to focus on the animal cells. And I like to, let's see here, how do I want to do this? I like to walk through the process of, for example, protein synthesis, before we go through and look at like each organelle separately and talk about what it does, because I think it helps if you know the story of what these different components are doing in the actual production of a product. I think it might help you remember them better. And then maybe as we're going through, you can kind of see if you have questions with that. But so this would be an animal cell. And here in the center, so this is the pink here is the plasma membrane. That's the outer plasma membrane or cell membrane. That's that phospholipid bilayer. And then this purple circle here is going to be our nucleus, which is where eukaryotes have the DNA separated by this nuclear membrane, which is also called a nuclear envelope. Glad I noticed it called it something else, because there's another one of those where you might see different terms. But that's also a phospholipid bilayer as well. And the kind of blue hazy circular structures that you see in here are actually the chromosomes, and we refer to these as chromatin because chromatin is the DNA and its associated proteins. The proteins help to allow us to, so the DNA is a super long strand. And when we get to the next chapter that we're going to cover, we're going to talk about how the cells replicate. All that DNA has to be copied, and then it has to be pulled apart into 2 cells. And if we didn't have these proteins that the DNA was wrapped around in order to package them, it would get all tangled and it would be breaking and it would be a mess. So the chromatin is the DNA and its associated proteins. I didn't mean for that to be purple, but whatever. So if you see that term, it's referring to the DNA. So those are the chromosomes in the nucleus. And we'll come back to the nucleolus here in a second, but actually I'll just go ahead and say it now. The nucleolus is the structure or the organelle that produces ribosomes. I'll just put this in a different color so that it produces ribosomes. So the ribosomes are actually made at the nucleolus and then exported out of the cell, out to the cytoplasm where protein synthesis takes place in the cytoplasm. I do not like this new annotate. I have to click like 5 times more to get what I want to accomplish. Not cool. Okay, and the reason I say that is because we're going to talk about ribosomes here in a second. So that's where the ribosomes are made. Then those subunits are exported out of the nucleus. These little red dots out here are free-floating ribosomes. You can see kind of one here in the cytoplasm. And then there are also some that are attached to the rough ER, which is why it's called the rough endoplasmic reticulum, because it has those ribosomes on the surface, and those, when you look at it through a microscope, it makes it look rough. So that's where proteins are made, either free-floating ribosomes in the cytoplasm or attached to the endoplasmic reticulum. So we'll just follow, let's just say an example of insulin production. So we're going to produce the protein product, insulin, the hormone. So in the nucleus, there is DNA that is the gene that codes for, gene is a specific section of the DNA that has the information or the recipe for a single protein. So we're going to say in the DNA, we have the gene that codes for insulin. And because the DNA is, oh great, I put it in a color that doesn't really show up, so. I'll just put it up here, maybe it shows that a little bit better. Because the DNA strands are so long, those chromosomes are so long, the DNA is locked in the nucleus. And so we need to get that information, that sequence, that recipe for that code for insulin out to the cytoplasm, to the ribosomes. So in order to do that, the DNA is going to be transcribed. We'll talk about this process later, but the DNA has to be transcribed into messenger RNA so that it can go out to the cytoplasm. We abbreviate it as mRNA. You can kind of think of it like the DNA is in the recipe book, and the recipe book is locked in the cabinet in the kitchen. But we need to get that recipe out to the kitchen so that we can read the recipe and use the, the blender and know all the ingredients we need to add together and everything to produce the protein product, to produce the cake in my example. The messenger RNA copies just the information from that single gene for insulin. So that messenger RNA is then going to carry that information that we just copied from that DNA from the nucleus out to the cytoplasm. So we copy the DNA into messenger RNA, and then now that messenger RNA can leave through a nuclear pore to carry that recipe card. It's like we took just that recipe on a little recipe card, just that one, and we're taking it out to the kitchen so that we can build the cake. So we have to bring it out of the nucleus, out to the cytoplasm, so that it can go to a ribosome in order to build the protein product. The ribosomes that are attached to the rough ER are largely going to be producing proteins that need to be exported out of the cell. That's why they're there, because they're going to get packaged in a little vesicle that can carry that protein out of the cell. The ribosomes that are free floating in the cytoplasm are going to be making proteins that just stay there in the cell. So the enzymes that need to catalyze reactions within the cytoplasm and whatnot. So now the messenger RNA has gotten to the rough ER, and we're going to make the protein product. So now we make the protein, which in this case, in this example, is the insulin. And now once the insulin is made, that insulin is going to travel via a vesicle to the Golgi apparatus. The Golgi is going to function, I'll put this down here under the Golgi. It functions to process, package, and modify the products that are made from both the rough ER and the smooth ER. So process, modify, and package. I like to think of it as the UPS of the cell, or the FedEx, or the Amazon, whatever you want to call it. So that puts it in vesicles that contain receptors on them that say where it needs to go, whether it needs to be secreted out of the cell, or whether it needs to be transferred somewhere else in the cell, where it needs to go once it leaves the cell. So that's what the Golgi apparatus does, is it says, okay, now we got this protein, got this insulin, and now I need to tell everybody, tell the cell where it needs to go. So that's what the Golgi apparatus does. And then the vesicles that are going to carry that out of the cell would be like these little structures here. And all of these that are membrane bound, the rough ER, the Golgi apparatus, the vesicles, these all have that same phospholipid bilayer structure that forms the membrane that holds that stuff in there, whatever it's carrying. So that's those main organelles. The other ones that are not directly involved in that process of protein synthesis that I just talked about would be the mitochondria, for example, which hopefully you know about that because we talked about the whole entire cellular respiration chapter, but those are the ones that carry out cellular respiration that make ATP.
Mm-hmm.
And then lysosomes are just specialized vesicles that stay right there in the cell. And those are what we call the recycling centers. They have enzymes that break down old worn out cell parts or anything else that needs to be recycled and broken down. So do you have any questions about that process? Because then I'll go to the other ones here in a second.
It's really complicated. There's a lot of steps to that, but it all makes sense. Like,
yeah, it's just a matter of remembering the names for them and which one, what job each one does.
Right.
All right, so let's go on to pull up real quick. I can't remember if so other than for the photosynthesis chapter, there's nothing specifically from this chapter that you need to know about the plant cell. So I'm just going to kind of skip over the plant cell. And then if something else comes up about it, we'll come back to that. But a lot of the details for the chloroplasts in the plant cell, those are the ones that carry out photosynthesis that produce sugars from sunlight. That's kind of a separate chapter. So we'll just focus on the animal cell structures for right now.
okay.
All right, so this is just kind of reiterating what I just discussed on there. The chromatin is the DNA and its associated proteins. We'll come back to that in the next chapter as well that we get to, that's after the quiz. And then I mentioned that the genes are a section of the DNA that corresponds to the information to make a single protein that has to be copied into messenger RNA to get out to the cytoplasm. The nucleolus is where the RNA is made. There's so messenger RNA is the one that's the copy of the gene, the DNA gene sequence. Ribosomal RNA is RNA that is actually a part of the ribosome organelle.
And they're separated by that lowercase letter there.
Yeah, it's just kind of an abbreviation. So it stands for ribosomal RNA is the part that makes up the ribosome. Messenger RNA is the part that makes up or that copies that DNA sequence. And then we talked about the nuclear envelope. It has the pores that the pores are basically kind of like the gatekeepers that regulate what can enter and leave the nucleus. That whole structure, it mentioned at the beginning, the endomembrane system, the nuclear membrane is actually that phospholipid bilayer is continuous with the rough endoplasmic reticulum, and that's continuous with the smooth ER. And then also the Golgi and the lysosomes are all considered a part of the endomembrane system. And that's just all because they're all enclosed by that phospholipid bilayer. All right, and then we talked about the fact that ribosomes make up proteins. Some are free floating and some of them are attached to the ER, endoplasmic reticulum, which is called the rough ER. And I'm going to decide how much detail we're going to go into protein synthesis. Actually, let me look real quick and see. I can't remember. Of course, it's just by title. Oh, here we go. I got it. Okay, we do have a whole chapter where we go in detail into protein synthesis. So just kind of store that on the back burner and we'll go into more detail later. Okay. But I think there is a section in here that you need to be familiar with, because I did say know the different types of RNA and what their function is. So let me see if that's listed in here. So we talked about the fact that the rough ER, it carries out protein synthesis, and that's because the ribosomes are studded on the surface. The smooth ER makes lipids. So that's another one that you need to know for the smooth ER. And then actually I'll add this in there because I can't remember if this is on there or not, but it also serves to detoxify certain chemicals that are taken into our cell. So that's the distinction between the rough ER and the smooth ER. Rough ER makes proteins because it has ribosomes on the surface. Smooth ER makes lipids and detoxifies the cell. Showing you all of that in a little bit more detail. And then, like I mentioned, the Golgi apparatus, which is sometimes called the Golgi complex, or it's sometimes called Golgi bodies. It's all the same thing. So anytime you see Golgi, you should think modification, packaging, distribution for that. And then, like I said, the lysosomes are specialized vesicles that have those enzymes that break things down that just stay in the cell. Vacuoles are in plants for the most part. It does say that there are some in adipocytes, which would be fat cells in animals that store lipids in them. But basically vacuoles are going to be storage vesicles for different things depending on where they are. Then we talked about the mitochondria produce ATP. One of the things that you do need to know that a lot of students get confused about is that mitochondria are found in both plants and animals. So plants have chloroplasts and mitochondria. They carry out photosynthesis and cellular respiration, whereas animals do not have chloroplasts. We only have the mitochondria that carry out cellular respiration, which is to produce ATP. And then, let's see, another point, I don't remember if this is a question, but may come up that you might need to know is that both chloroplasts and mitochondria contain their own DNA and ribosomes. So sometimes they're called semi-autonomous organelles. They're the only other organelle that contains its own DNA.
That's kind of cool.
Yeah, it is. And actually, well, it's a whole nother tangent, but it is believed it is believed that the mitochondria and the chloroplasts were originally bacteria, prokaryotic cells that were engulfed by another cell, and that's what started the development, the evolution of eukaryotic cells. Because they were like, this is kind of handy. I can make my own food and I can make ATP, and this is cool. So they evolved into it, which is that fact that they have their own DNA that's separate from the chromosomal DNA is evidence that supports that idea.
Right. That's cool.
So chloroplasts also. And then here, because we did kind of rush through that chapter 2. Okay, yeah. So for even the photosynthesis chapter, the chapter 6 material, you don't need to know the details of the structure of the chloroplast. You just need to know that those are implants. You do need to know the steps of photosynthesis and what are the substrates and products for each step, like we went over in class. And I actually shared, let me change my share real quick, and I'll just pull it up because I can't remember if you said you saw it yet. Which one? Nope, this one. That's on the quiz 1 study resources.
Mhm.
I shared the answer key. I also shared this in the discussion week 3 class and study activities discussion board, but I wanted to make sure everybody saw it. So I copied it into the study resources also. But this
Okay.
has that labeling answer key to the ones that I had you do in class. I also have the cellular respiration one on there. And I'm working on, I saved a video from another session where I actually walk through filling these out and explain everything. And then I think I also have, I haven't gotten that far yet because my computer was going too slow, but it also has the photosynthesis and cellular respiration cahoots. Can't remember if we did the photosynthesis one that I walked through in class with the explanation. So I'm editing that video that I'll also add in here in the study resources, and I'll send another message once I get that added in case anybody wants to watch that. It's a full class, so it's like 2 hours long. So I've got to find the parts that are going to help you guys with the explanation.
Nice. Well, thank you for that.
So that you don't have to watch a whole 2-hour video to get the information. All right, let's see here. So let's continue on. We talked about mitochondria. The cytoskeleton is the other group of proteins and organelles within the cell that are structures that you need to know that were not really, we didn't talk about them in that process of the insulin production. So the cytoskeleton is protein filaments that basically help the cell keep its shape. And then also a lot of these cytoskeletal proteins connect a cell to an adjacent cell to form the tissues. And then they also function for transport within the cell. So this is going to be these proteins down here, myosin, kinesin, and dynein. I can't remember. I don't know. I think I have it listed on here necessarily in the study guide. Trying to look real quick at my list. It might be on the midterm because I remember somewhere specifically there was one of those motor proteins that were referred to by name. I don't have it in this quiz 1 study guide, so it might be not until the midterm study guide, but eventually you'll need to know these. So I'm going to go ahead and just share that now anyways. These motor proteins basically act like the myosin is kind of like cables through the cell, and the kinesin and dynein act like a little gondola or car that's riding along that cable to carry things where they need to go into the cell. So you can kind of think of it like a little transport system. So the kinesin and dynein are both involved in actually carrying it's actually this one here. They're actually involved in carrying vesicles to where they need to go in the cell along the microtubule. So the microtubule would be like the cable. And then the kinesin and dynein are little proteins that use ATP. Every time they spend an ATP, it actually, I'm trying to put my hand in front of the thing, makes the the protein structure change shape, and it really literally looks like it's walking along the microtubule to carry the vesicles throughout the cell. It's pretty cool. They look like they're living, but they're just proteins that are changing their shape as they burn ATP, and it causes them to walk along the microtubule to carry the vesicles where it needs to go.
That is actually really cool.
I have a really cool, actually, I'll see if I can find the video. I used to show it in my class all the time. It's like a short little one that's an animation showing the process. I'll share that in the study resources also, because it's pretty cool. And then as far as the specific names of the cytoskeleton, I don't think you need to know the difference between those for right now, but there's basically 3 different types. There's microtubules, which are the largest ones in diameter. There's the intermediate filaments that are called that just because they're kind of medium sized. And then there are the actin filaments, are the thinnest ones that you may remember from the skeletal muscle system. Actin and myosin are the 2 protein fibers in muscle cells that slide past each other to contract a muscle. So that's just showing you the actin filaments. Those are the thinnest ones. And then centrioles, I can't remember if I have centrioles listed in here, but we're going to come back to them. We're going to talk about them in either the next chapter or the one after. So I may as well just say while we're here what they are. They're made of microtubules. These are what are going to form the spindle fibers, which are protein fibers that separate the copied DNA after it's copied during meiosis and mitosis. So just kind of store that one in the back of your mind, and maybe you'll remember what it is when it comes back up later. And then cilia and flagella, like we said in class, the only human cell that has flagella are sperm cells in the male, but cilia are also made of microtubules, and cilia and flagella both function to move things, just in a different way. All right, we're going to skip all that because you don't need to know that. And then this diagram is kind of nice because it's just showing you these different protein fibers. So collagen and elastin are 2 different types of proteins that form these kind of cable-like structures that give, this is the extracellular matrix. So this is outside of the cell. These protein fibers are kind of like this net that holds everything in place. And then this would be the phospholipid bilayer plasma membrane of an actual cell. And you can see there's proteins that cross across there that can connect to these other proteins to hold it in place and provide its structure. And then we already talked about the fact that the cell wall is an additional protective layer. Again, animal cells do not have a cell wall, but plant cells do, fungi do, and some protists do as well. And then also bacteria. Talked about collagen and elastin. And then these are just the different types of junctions between the cells. I can't remember if I have a section on there. Seems like I remembered there being something about that, but it might just be on the midterm or the final exam. But there's 3 different types of junctions that are listed here. I'll just kind of go over each one. Adhesion junctions are, so this would be one cell membrane, and this is the adjacent cell membrane. And adhesion junctions are proteins that extend out that are holding those 2 cells together. The adhesion junctions have a little bit of play. So these would be between cells that have to move a lot. So anything that has smooth muscle, like a vessel or something like that, or muscle cells, there's a lot of movement with muscle contraction. They need to be able to hold the cells together, but they also need to allow a little bit of movement. So that's the adhesion junctions. And then I made it too big. Now I can't see where the other ones are. Oh, okay, they're both on the bottom. The tight junctions are waterproof junctions. So these are going to be between cells that need to carry a liquid so that the liquid doesn't pass out from those. So for example, I was just talking about blood vessels. This would be another type that would be in blood vessels or capillaries the epithelial cells that form the vessel are held together by these tight junctions, because it wouldn't be very effective if the blood just leaked out between the cells instead of carrying it wherever it needs to go in the body. Same with in the intestinal wall. It would be not very cool if our feces being formed was leaking out into our abdomen. So these tight junctions keep those fluids that are inside there and other substances from leaking out. And then your gap junctions are going to be protein channels, basically, that form a tunnel between adjacent cells. I don't know if you can see that in this picture, but it's actually, this is one cell here, and this is an adjacent cell here. And these proteins are the blue structures that form this tunnel that allows the passage of things like ions, so things that can't pass across the plasma membrane. There are a bunch of gap junctions. You may remember this from cardiac muscle, from A&P, that the intercalated discs contain these gap junctions that allow for the passage of ions that initiate the muscle contraction in the chambers of the heart, so that they can all, atria all contract at the same time. The ventricles all contract at the same time. So these gap junctions help to distribute those ions that tell the muscle to contract. Right, and I think that's it. That's in this chapter. There's a lot more slides, but these slides, for whatever reason, have extra stuff at the end. Yeah, so that's the end of it.
Okay. I feel so much better about the chapter.
Perfect. All right, so do you have any other questions?
I don't think so. I'm just gonna go through and fix all my typos here because there's a lot of them.
And I'm gonna like I said, I'll edit this video and the other video and post those in the study resources, but I was gonna, especially since we kind of rushed through the end of those last two chapters, I'm gonna give just a little bit of time in case anybody has questions at the beginning of class. So if you come across any questions as you're studying, you can either send me a message with those, or you can bring them to class and I'll help you out.
Sure. Well, I genuinely appreciate your time amongst the craziness of
Absolutely.
everyday life. And I feel much better about the chapter now. So hopefully that